DNA from a chemistry perspective. So DNA stands for deoxyribose nucleic acid. It's a polymer, so it's a large molecule. It's double-stranded. It contains complementary base pairing. Where A binds to T, C binds to G. Remember the A stands for adenine, C stands for cytosine, G is guanine, T is thymine. Now if we check out this page of my biology guide actually, but it does show the structure of DNA really nicely. So we said that DNA is double-stranded. Now the backbone of the DNA is made up of phosphate sugar alternating. So there's the phosphate, here's your deoxyribose sugar, and then the two backbones are linked by the rungs of the ladder, which are the complementary bases I've mentioned, where A binds to T, C always binds to G. Now these bases are held by hydrogen bonds, 2 hydrogen bonds between A and T, 3 hydrogen bonds between C and G. One thing to be aware of is the term nucleotide. All a nucleotide really is the combination of a phosphate, a sugar and a base. So there's one nucleotide, here's a second nucleotide. So now let's look at all of these various structures from a chemical point of view. So we're going to start with the structure of phosphate. Now handily this is included on the data sheet, so don't worry about learning these, but I just want to show you the structure here. What about the structure of your deoxyribose? And technically speaking, it's actually 2 deoxyribose. So the ribose refers to the fact that you have a sugar made up of 5 carbons. It's a pentose sugar. Here's its structure. As I've already said, nucleotide consists of a phosphate joined to a sugar and then a base. So in simplistic terms, something like this. There's our deoxyribose sugar. So let's combine this simplistic diagram with the actual structures. So we're going to start with our phosphate. And then up here you'll find your base attached. I'm just going to number these carbons. This is carbon number 1, 2, 3, 4, 5. You'll find that your next nucleotide is attached up here. And your next nucleotide is attached down here. What about the structure of these bases? As I've already said, we know that DNA is made up of 2 strands that form a double helix shape. I've said that there are hydrogen bonds between the complementary bases. And as we see here, we know that adenine binds to thymine. I said that there's 2 hydrogen bonds, and we can see how that forms between the oxygen here and the hydrogen here, the nitrogen here and the hydrogen here. And then what about in guanines and cytosines case, where we have 3 hydrogen bonds? Happily, you're given the structure of these bases in the exam, but it is important that you rotate them in the correct orientation to show how the hydrogen bonds form. The anti-cancer drug cisplatin operates by reacting with the guanine in DNA. Figure 1 shows a small part of a single strand of DNA. Some lone pairs are shown. Remember, this is the phosphate that I was just telling you about. Here's our deoxyribose, forming the backbone, phosphate sugar, phosphate, and here's our guanine base. The DNA chain continues with bonds at X and Y. State the name of the sugar molecule that is attached to the bond at X. Yeah, we just need to name that. That's 2-deoxyribose. Messenger RNA is synthesized in cells in order to transfer information from DNA. The bases in one strand of DNA pair up with the bases used to synthesize RNA. Figure 2 shows 2 bases used in RNA. Suggests which of the bases A and B forms a pair with guanine in Figure 1 when the messenger RNA is synthesized. Explain how the base you've chosen forms a base pair with guanine. So I'm just going to start by writing that it's going to be 3 hydrogen bonds. Let's identify where that will be, and therefore whether it's base A or base B. So I'm just going to bring them up so you can see them more clearly up here. So we need to work out where our hydrogen bonds will form. So I've moved up base A here to actually show you nice and clearly, but obviously this represents the lone pair of electrons. Now you can see here that a hydrogen bond will form between hydrogen and this highly electronegative oxygen. Hydrogen bond here between this hydrogen and nitrogen, and then this oxygen and this hydrogen here, which is why it's base A. And as we'd expect with guanine, it will be 3 hydrogen bonds, so this is likely to be cytosine. So 3 hydrogen bonds form between base A and guanine. And actually, the way I've done it here, rather than writing a big description, you could just redraw the structure and show the hydrogen bonds, and that would be a clearer way for the examiner to see your answer.